Okay, let's continue with our discussion intermolecular forces. So basically, your intermolecular forces are attractions between molecules that help maintain biological structures. Please do remember that it helps them maintain biological structures. So ginagamit siya, the function of your intermolecular forces is to help those biological structures para maging stable, to become stable. Number one here, we have your hydrogen bonds. Your hydrogen bonds is the weakly attraction between hydrogen and electronegative atoms. The common atoms we have the oxygen and nitrogen. Example of this again, it helps to maintain biological structures. Example of this, it stabilizes the DNA structure and helps maintain protein shape. So we have the pictures here. As you can see, we have the positive and the negative pole or the molecule. So dito, sabi niya dito, the positive pole, one of the molecule is attracted to the negative pole of the other, forming hydrogen bond. So here, we have the negative pole and we have the positive pole, forming a attraction between the positive and the negative molecule. Again, it forms your hydrogen bond. The simple it is. Diba? Next, we have your hydrophobic interactions. Your hydrophobic interactions occur when nonpolar molecules avoid water, meaning water feeding. Diba? Hydrophobic. Phobic means natatakot, ayaw niya. Water feeding yan. Molecule clusters together. Examples, we have the lipids forming cell membranes, and importance, help organize biological membranes. So we have here the water molecule and the hydrophobe. The hydrophobe, as you can see, hindi nagdidikit. It cannot touches the hydrophobe with your water molecules. Okay, next, for your electrostatic forces or the ionic interactions, your ionic interactions, attraction between opposite charges. Examples, the positive amino groups interacting with negative carboxyl groups. Importance, it stabilizes proteins and molecular structures, parang si hydrogen bonds. So, makita mo dito yung kanilang sodium and chloride. Again, they are the positive molecule and the negative molecule. Kapag nagdikit yan, we have your electrostatic force, your sodium chloride. For your van der Waals naman, van der Waals forces, weak attractions between nearby atoms or molecules, importance, helps molecules maintain proper shape. So this is your van der Waals force. Next, for the covalent bonding, a chemical bond form an atom share electrons. So it can share your electrons. Types, we have your polar covalent bond. Electrons are shared unequally. Example, the water molecule. So dito, makikita nyo, as you can see here, we have the two hydrogen molecule, tapos dumidikit siya dun sa oxygen. So, iisa lang. Yung sinasabi niyang unequal kasi, as you can see, there is only one molecule of your oxygen. And there is two molecule of your water. So, there is an equal electrons shared. Ayan siya. For your nonpolar naman, electrons are shared equally. Example, many carbon-carbon groups. Example here, we have the unpaired valence electrons and an equal sharing of valence electrons. So, pag nagdikit yung dalawang yan, makikita nyo dyan, we have your two molecules here, the two molecules here, they have equal sharing of electrons. Okay, let's now proceed for your chemical reactions in biochem. Number one, we have your dehydration synthesis or the condensation reaction. Please do remember that in your condensation reaction, it is joined together by removing water. The function of this, it is build larger molecules from smaller units. Examples here, yung lagi kong sinasabi, the most common examples I have talking last week, we have your amino acids. That your amino acids, when amino acids forming amino acids form into your proteins. So from your smaller units up to your larger units. For the monosaccharides, it becomes polysaccharides. For next, we have your esterification. Your esterification are reaction between an alcohol and an acid that produce an ester. Example, we have your glycerol plus fatty acids. We have your triglycerides. That is your esterification. For the hydrolysis reaction naman, a reaction where water is added to break large molecules into smaller units. Function, it breaks polymers into monomers. So it can break your polymers into monomers using your water. From the word itself, hydro means water. Hydro means water, and lysis means breakdown. So hydrolysis. Example here, we have your proteins using your water. It can break your proteins into your amino acids. For your carbohydrates, it can turn into a simple sugars. Next, for the self-regulation of cell reactions. Cells perform thousands of chemical reactions every second to maintain life. This reaction must be controlled and regulated to prevent too much or too little activity. The cells regulate their reactions mainly through the action of enzymes and enzyme control mechanism. Your catalyst is a substance that increases the rate of speed or chemical reaction. It works by lowering the activation energy needed for the reaction to occur. For reaction to occur. For the activation energy is the minimum amount of energy required to start a chemical reaction. Please remember that your catalyst, it is a substance that increases the rate or speed. So, pinapabilis niya lang. Pinapabilis niya yung isang enzymatic reaction. Without enough activation energy, the reactants cannot break their existing bonds. New products cannot be formed. Ganun lang. So, it requires your activation energy in order for enzymatic reaction to start. Example, a chemical reaction may occur slowly without a catalyst. When a catalyst is present, the reaction happens faster because less energy is needed. Your enzymes, an enzyme is a biological catalyst. Most enzymes are proteins. Again, most enzymes are proteins. Enzymes speed up chemical reaction in living organisms. Functions of enzymes. Number 1, we have accelerates metabolic reactions. It helps build and break down molecules. It can build and it can break down molecules. And number 3, maintain normal cell activities. How enzymes works? The enzymes lower the activation energy required for reactions, and they do not get consumed during the reaction. Hindi po sila nako-consume. Please do remember that. They are not consumed during the reaction, and they can be reused. Pwede silang paulit-ulit na gamitin. For the collision of reactants, the reactant molecules must collide with, number 1, enough energy. So, kailangan nila makipagbanggaan with the enough energy. The proper orientation to allow chemical bonds to break and new bonds to form. So, it requires the enough energy and proper orientation. If collisions do not have enough energy, the reaction will not proceed. So, it must, kailangan meron kang enough energy in order for the reaction will proceed or will start. Okay, next. The enzyme and substrate complex. The molecules acted upon by enzymes are called substrate. And steps, the substrate binds into the enzyme and the substrate attaches to a specific region called the active site. Sa active site lang sila magbaband. Doon lang sila mag-attach. It will attach to the specific region called the active site. And the enzyme substrate complex is formed. So we call it the enzyme substrate complex. The enzyme converts the substrate into the product and the product is released and the enzyme can be used again. Ito yung sinasabi natin kanina that hindi siya nagagamit, hindi siya nababawasan, but it can reuse over and over again. So here we have your substrate. We have your active site. We call it this active site. So this is both the substrate. So here we have the enzyme and substrate complex. Makita nyo dyan. So again, specific lang po yung pinupuntahan ng mga substrate. It has a specific active site na pupuntahan. We have here, for example, this is a semi-oblong and this is triangle. Well, basically, for the enzyme, kailangan, yung enzyme niya, kailangan specific lang sa kanyang substrate. So here, the enzyme product is complex. The product here, we have your product. And enzyme plus product leaving active site. So this is your product already, and this is your enzyme, and the product is leaving active site. So it will, this is the lock and key hypothesis. For the induced fit model, we have the substrate, the active site here. So makikita nyo dyan for the substrate, for the enzyme, hindi na specific yung shape niya. The enzyme changes shape slightly as substrate binds. So sabi niya, nagbabago daw yung enzyme, it will change your enzyme according to your substrate. So as you can see, nagbago nga. It changed already. From here, it will change according to your substrate. So here in the products will release. Okay. The active sites, the specific enzyme or the substrate bind, characteristics of your active site has a specific shape, matches the shape of the substrate. Well, according to your lock and key, it has a specific shape. But induced fit, according to induced fit model, your enzyme will automatically change if your substrate already bind to your, or your substrate will bind to your enzyme. For the lock and key, I already discussed that. For the enzyme example, we have your carbonic anhydrase. Your carbonic anhydrase is an enzyme that speeds up the reaction involving carbon dioxide and water. So using your carbon dioxide and water, it can speed up the reaction. And we call it your carbonic anhydrase reaction. We have here carbon dioxide plus water. We have your carbonic acid. Importance. It helps regulate carbon dioxide levels in the blood. Important in respiration and acid-base balance. Without this enzyme, this reaction would occur very slowly. So using this, it can hasten your reaction. Your carbonic anhydrase can increase the reaction rate millions of times faster. So ganun siya kabilis. And regulation of enzyme activity, cells control enzyme activity to maintain proper metabolism. One way cells regulate enzyme is through enzyme inhibition. Okay, for your enzyme inhibition occurs when the molecules decrease or stop enzyme activity. So the molecules that reduce enzyme activity are called inhibitors. So it has your inhibitors, types of enzyme inhibition. Number 1, we have your competitive inhibition, non-competitive inhibition, and irreversible inhibition. For your competitive inhibition, makinig, a competitive inhibitor blocks the enzyme active site. Balikan natin yung active site. We go back to your active site. Again, this is your active site. So, again, for your competitive inhibition, it will block this. It will block your enzyme. So, if this one is blocked already, is your substrate will fit into your active site? The answer is no. So, that's why that is your competitive inhibition. For your non-competitive inhibition, the inhibitor does not bind to the active site. Instead, it attaches to the another part of the enzyme. In fact, the substrate may still bind, however, the enzyme changes shape and cannot properly perform the reaction result. So dito sa non-competitive naman, anywhere for your non-competitive, anywhere it can bind. For example, the substrate will bind here even though this is not an active site. That is your non-competitive inhibition. For the reversible inhibition, the inhibitor permanently stops enzyme activity. It usually forms a strong or covalent bond with the enzyme. Effect, the active site becomes permanently blocked. So, permanently blocked yung sa kanyang irreversible inhibition. Importance of your enzyme regulation. Enzyme regulation allows cells to prevent unnecessary reactions, control energy use, maintain balance in metabolism, respond to changes in the environment. Without enzyme regulation, reactions may happen too quickly. Important molecules may be depleted and cells may become damaged. For the summary, catalysts speed up the chemical reaction and the enzymes are biological catalysts that control cellular reactions.